आप देखिए वहीं से हो गया तो फिर बस यहीं तक सामने रखना पड़ता है
years ago, so that's the big anniversary this year. It's funded by EMBO member states, so there's 31 EMBO member states directly contributed to the European Molecular Biology Organization, but there's also associated states, and India is a big contributor is one of the associated states, which means that all of you can be part of EMBO activities, which are many. You can start when, you've all gone beyond the time of fellowship, but you can tell your students that they can apply to EMBO fellowship and also get money to travel across the entire world. Fellowship also, you can work wherever, as long as you're part of the member states or be associated to EMBO. EMBO also, you probably know its activity in course and workshops and lectures. And also EMBO elects not only its members, but also it elects young group leaders at the part of the EMBO Young Investigator Program. I was part of both. I started with them as YIP. I was in 2000, and I became EMBO member in 2017. You can also participate to EMBO activity by publishing. So we heard about, open science. EMBO is a big player in terms of open science. It started a new form of reviewing papers in Review Commons, and I encourage you to look into this. It's, my lab has used it. It's very useful. It makes publishing a much happier experience than the traditional way, because as soon as your paper enters the streamline of Review Commons, you know that it will come out and you will be published in good journals, because there's a portfolio of journals that not only include the journals that depend from EMBO listed there, but also a whole portfolio of EMBO which basically embrace the same philosophy of publishing. So now we'll just start my talk by just talking about the journey I made. And so yes, I'm part of the international program because I traveled a little bit across the world during my research life. I did my PhD, so I'm French originally. I grew up into this tiny speck of country which is just at the border on the Atlantic Ocean between France and Spain. It was a tiny town. By Indian standard, 20,000 inhabitants. It's not even a town, it's nothing. No, but in France it's a town. And but I studied in Lyon, and then I did my PhD in Plymouth. I did a short postdoc in another part of England, in Norwich, and then got the first position in Lyon. That was a permanent position. French system starts like that. After 1 year, you're permanent and you get your position for life. So it's just like getting married to an institute. At that time, it was really like an arranged wedding. So I got this position because I was more or less invited to apply by someone I could not say no to, because I knew that I would not be able to get a position if I somewhere else in France. So I was a good boy, but once I got there, things didn't work out very well. I had been being educated in the English system. The French system is more or less the exact opposite, and I could not really fit. So within a few years, I realized, okay, that's not for me, permanent, okay, but I don't like that. And by serendipitous visit to Singapore, where I replaced on short notice a colleague who could not go there, sometimes bad is good for you. They had the SARS in Singapore, and this caused one of my colleagues with respiratory problems to say, no, I can't go. It was 2003. Went there. It was a new institute called Temasek Life Science Laboratory, and they told me, we're looking for people. Would you know people who could be interested? I say, me. And about 6 months later, I told my lab, we're all moving. That was a time that was absolutely fantastic. I spent 10 fantastic years in Singapore. And for various reasons, we don't have time to go into this, I decided to get another position, and I got this position in Vienna. So going back closer to home. That was one of the reasons that made me go back. And then I became senior PI there. So this traveling, and that's a point I need to emphasize. Traveling is causing you to change because you have no other option, but also because you're exposed to a completely different environment. And this is reflected by the change in kind of sort of expertise and types of work I've been doing in the past 25 years of research in my lab. And this stands also in part with changing model. So I started with a non-model species, which is a brown alga called Fucus, that was a historical model to study polarity. And then I realized that was the rise of molecular biology and genetics using a model called Arabidopsis, this tiny plant that's relative of cabbage. And so I said, okay, after trying hard with Fucus, which led to interesting results, I will just switch and jump on the bandwagon on this Arabidopsis research, which I carried to Singapore, but then became another model started to be available, which was moss, or a relative of a moss called Marcantia. It actually grows probably in the waterfalls close to Bhopal. And going back to India, we started to, as you will see, work with far more models. And this was led by the research environment around me. So basically, that's, I think, what would be perhaps the kind of thing you would think you should do when you start a research job. Define broad questions. Surf on new technology, be aware of what's up and coming. Don't stay, in the old ways, and choose the right place. As I explained to you, I did none of that when I got my first position. That was certainly not the right place for me. So you have to harness eventually serendipity, and that's what led me to Singapore. And then we went to this initial work in France. So what people told me in France when I arrived, you have to work on something useful, which is this endosperm. That's what you eat, that's the rice, that's wheat. So that's very important. And there had been, in fact, very little genetic research on endosperm. And I said, yes, endosperm, yes. And they said, but you have to work on maize. And I said, no, Arabidopsis is just up and coming new model. It's going to be faster. And they said, you have to work on maize. So I said, endosperm, yes, maize, no. And then they said, it's like that. So if you want to work on Arabidopsis, you're on your own. And they basically decided not to fund any of my research for quite a while. So that's when you have a divorce, between the funding body, which is just stupidly top-down. Top-down does not work to do good research. It has to be bottom-up. And then after a while, but all bad things have a good end if you just take it correctly, because working on endosperm led me by chance during doing genetic work to realize that some controls of endosperm development had a very weird genetic regulation, which was purely maternal. So we found first mutants that exerted a maternal control on seed development, on embryo development. And this led us to, in fact, start to work and discover with another lab in Switzerland and a lab in Australia, working at the same time through different routes. Our work all converged to define this molecular nature of this maternal control, which was imprinting. And this led gradually to the discovery of more and more imprinting genes. And that was the initial research focus I had when I moved to Singapore in this building in TLL. And that's when I built really my interest also in chromatin, because basically the genes are the maternal control in the seed development is due to the fact that the paternal, only the maternal copy of a gene is expressed and the paternal copy is silenced. Hence, This gene is going to control basically all its product from its maternal copy, and that's why you have imprinting. Imprinting is also found at that time. Imprinting was not only in seed plants, and was further found in all kinds of seed plants, but was also known to be found in humans and in all vertebrates, in all placental mammals. So, so there was an interesting convergence between the two fields, and I actually organized an EMBO an EMBO conference when it started to be possible to organize EMBO conference outside of Europe, in Singapore, on imprinting. And doing that as relatively junior PI was really important. So I encourage you, use EMBO that allows you to organize conferences in India, and there's many EMBO workshops that have been organized to network. This is, you know, in addition to India Bioscience, and India Bioscience collaborates with EMBO in organization of those meetings. It's extremely important. It puts you on the map, and people cherish for years the memory they have of these EMBO meetings you organize. Here they had even more memory because I organized the breakfast in the jungle. So, Singapore has not much nature, but it has a little bit of secondary forest right in the middle of the city that you can access in the morning and go and have breakfast after you go and bring people on the equator. You know, most people coming from the north, they don't know, they have no idea of anything, and they really marvel about the whole thing. Except that after half an hour in the forest, we had these torrential rains that happened in Singapore, so everybody was soaked to the bones, and we had to rush to the hotel. I can tell you that nearly 20 years, I mean not 20 years, but 15 years later, some people, when I meet them, they remember, oh yes, remember this meeting. So, but that helped me a lot. Actually, it helped me when I applied for the next position. I could ask people who had participated in this meeting to write reference letters, and they were useful. Another kind of serendipity is when you meet people, when you go to, you know, to conference or talk, and you don't really, it's not your field. And there was basically the boss of Benjamin Lopin gave a talk on Drosophila early development when I was still in Lyon, and I attended this talk, and I thought, oh, this pattern of development in what we're looking in early embryo looks very much to what we're seeing in endosperm. And I want to talk to those guys. I had no idea. And I went and I asked just for a slide to just give a comparable during training. And then the slide was given to me by Benjamin at the time. We became good friends. And at some point he needed to have a bit of help with accessing confocal microscopy. And I happened to be the head of a confocal microscope facility in another place in Lyon. So I told him, come along. And we started to basically work together on this confocal microscope. And the mutant he was working on, that is illustrated there, it's a mutant in Drosophila that causes the paternal genome not to be incorporated with the maternal genome. So the genome that arrives at the beginning, you see this kind of sort of red ball, that's the mother genome. And eventually, if the two genomes cannot incorporate together, the paternal genome is going to be eliminated. And this is because, it turned out, it's because, in fact, these mutants are deficient in chaperone that helps the chromatin to be incorporated back on the paternal genome after it loses its protamines just after fertilization. And this chaperone incorporates a type of histone which is an unusual, a variant of the canonical type of histone. And I became fascinated about that. And then I gradually focused basically all our work on these histone variants, and moved in Singapore, I discovered an histone variant called H2W that is only present in plants and is deposited on transposons and helps to build a chromatin form that traps transposons and maintain them silent. So that was, you know, a little meeting like that, a slide that fascinates you, and you just unload a whole kinds of career in a direction that you would not have envisaged. So plan on serendipity, be curious, go outside of your field of research because that's very helpful. And so when I went to Singapore, we had to negotiate a little bit, but I'm not going to talk to you about that because it was a very special institute at a very special time. When I moved to Vienna, I went through something which was more usual, and just things, everything, negotiate, secure everything before you sign. What you put your signature on, you won't be able to change anything, it's too late. Deal with national funding agency from the early stage, learn from your colleagues, of course. Challenge with working, with networking. You have, when you're a new PI, often it's quite difficult to network. I mean, you're busy with moving your team, especially when you do that in mid-career. You have to just deal with, I mean, I commuted between Vienna and Singapore, meaning that I was taking a plane going to Singapore every month, spending 1 month there and coming a plane back. And I've done that for 2 years. I actually lost my sleeping pattern for a long time. I don't recommend doing this, but I had to do this, and the team was emptying on one side, and I faced this challenge of networking there at the beginning because I was not present enough. That didn't prove to be too good, so that was a mistake I done. I mean, but I had not much of an over opportunity. But be very careful. When you arrive, you're the new guy or the new girl there, and you're interesting to your colleagues for a little while for the novelty. This interest is not going to last. So do your networking very early, because otherwise it's opportunity missed. And that's it. And yes, so it's very important that you negotiate when you move in mid-career. You're going to have to really take care of the people. I've done that twice in my life. And so you have to, one thing that is very important, and though you have to be adamant there, is if you move mid-career, you say, there's a non-negotiable condition. I need so many positions with me because I need to move my lab. You can't negotiate about that. And you have to put that on the table. So, and associate the people in your lab early also when you want to move. This is super important. They have to be part of your move. They have to feel that they're doing the project with you. Even before you do that, before it becomes official with your admin. The admin of the place you live need to be the last person to be involved, because they're going to be pretty evil with you most of the time. So, current research, I will be quite brief. Imprinting came back to the lab, and we're working on imprinting in bryophytes. Because we found that, in fact, bryophytes are these mosses. They also do maternal care of these embryos. The little embryos grow on the mother plant in the bryophytes we are working on. And in fact, they form a very drastic form of imprinting because they just shut down all the paternal genome during embryogenesis. And these plants that you see actually in the wild, they are haploid. All mosses are haploid. And when they get the embryo, it's the only time when they become diploid. In fact, they do all they can for the embryo to remain haploid. And we don't know why. But why they're doing that, the biological significance of that. But we know at least that this is brought about by the same silencing machinery that participates to silence your developmental genes, the genes that pattern you. And so we thought that we have to reconsider the role of this silencing machinery, which is called polycomb repressive complexes. And in fact, something that we realized was that we had to look back in time in evolution to start to understand what polycomb is really doing. And this completely changed our view on what polycomb is doing. We now have shown that polycomb is actually a machine which originally was there to recognize and silence the non-self, the transposons, but probably also all the genes that came in the genome through horizontal gene transfer. When we're doing that, that led me to really just realize I have to really go back on time. And basically, I will just skip that. And our current research is now really oriented towards evolutionary research. We'll just skip this. Yes. So hiring is a big thing. And I will just be brief. And when you start, it's something. I would say, some of the time, use your gut feeling. Use your gut feeling. You're going to have, especially as a young group leader, you're going to have to work with your lab members as, of course, you're their mentors, but you're very close to them. Things happen and they're also quite close to you because the age gap is not that big. And quite a lot of things are happening to you which are generally quite dramatic at that age, which is dealing with building families. And they're dealing with, building their future. And you're both dealing with complicated things. So you have to feel that you're going to hire people they're not your family. You don't have to be paternalistic with them. But at the same time, you have to really feel that you can work with them. Don't submit them to too many interviews and all kinds of things, but make them talk with your lab members, more than with you, actually. Because also they have to fit with the lab members. If you have no lab members, then talk perhaps with other PhD students in the neighbor labs. That can help. They need to feel that they have also chums, that they have friends, people that they will be able to relate with when things are going to become perhaps difficult with you. And that's what my main advice as a young PI is to use this. Strategies to ensure productivity and efficiency show that you care, and they will care for you. And that's all I could say. And the mentorship is something that is a never-ending learning curve. You will make mistakes, live with them. The best thing I heard from a director of an institute I heard was, I was wondering how, I asked him, I was a student and he was the director of a place that was building out of the ground where we were there. And I was really asking him, how do you manage, to take all these decisions all the time? And he told me something I never forgot. I managed to do that because I managed to cope with my mistakes. If I make 30% mistakes, that's fine. So accept to make mistakes. The people will see that you have to be humble, you have to make mistakes. Okay, that's okay. Don't try to be a superhero because you're going to just lose steam. And sometimes also it will make you feel that you can't accept, or people feel that you can't accept failure. So do so. That's the only little advice I would tell you after years. Okay, I will just script this. Be part of the other people to create a fair working environment. And this is a global trend now throughout the world. I heard the same thing from colleagues here. We have to take care of that. We have to take into account that, yes, people build their families, and you have to make people be able to have this balance between working and living, and with a special care for people, who have to take care of families more or less single-handedly, or when the two in a couple are also working. Okay, so just end up with very active, be an actor of the scientific community in your country, but also internationally. Okay, I've been traveling post-COVID. There's been my travel, when I started to travel again across the world, I could feel something that is not going perhaps so well. It's that a lot of countries tend to start to close on themselves. My last visit in China in September was really conveyed that so much. Where science works only on a global level. You cannot work science at the national level only. That will not work, even if you're a big country. Don't think you're ever self-sufficient as a country. You have to work for a global science community, travel as much as you can, work across. Do not forget, of course, your local environment. They're, of course, extremely important, but do not forget this international. And EMBO and India Bioscience can really help you, but also other actors. These are very good guys. Cold Spring Harbor, Asia, is trying to diversify the sites where they want to do their conferences. And I think it would be worth to approach them to envisage India. Company biologists can really help you. Gordon research conferences are really good. And of course, EMBO conferences, which are held in India, I co-organize some of them. So I will finish with this. Just got that. Just tell, yes, in terms of like life is short and the career will be flexible. You need to have this flexibility. Now we envisage in my lab to make a big move to start to work with eukaryotes, with archaea. And I think you have to be open to big changes in your life. You can't remain on the same topic for 25 years. This is suicidal. This is bad for the brain. This is probably triggering, early dementia. Don't do that. Innovate, be bold, think out of the box. And also one thing that is changing, and you're going to be the actors of that. I think this model of a single PI with a pyramid and all that, it's finished. It's going to be finished. Unfortunately, agencies don't know how to evaluate us in another way. And you have to make a big call to start to think about labs which have 2 PIs, labs which are temporary. That could be just a consortium of 2 or 3 PIs across different parts of the country that could work for 5 years and dissolve and reassemble. Think in these terms. The projects now need, you don't see a paper where you generally, comprehensive papers will have aspects of biochemistry. If you need cryo-EM to have your structure, you need genetics, you need perhaps even population genetics to make something which is coherent. Or you work with several models. So think about how you can use your local community in India to create this virtual lab and perhaps try as much as you can to use these meetings like that to make this message go bottom up, such that also the Science Foundation helps you to create funding that allows these virtual labs to exist. That's my contact if you ever want, but I'm here for the next 3 days, so take advantage of it. And thank you, and we have training you can read.
Hello. Yeah. Great talk, Frederic. Thank you. 1 or 2 quick questions. Yes, Andrew.
Frederic, if I can be a little critical, you are speaking from a sense of security which is not very easily available in the Indian environment. So I noticed that a number of your publications are in bioarchive. You put them up as preprints. How does this affect the careers of your Asian students, not the European students? Does it impact them in any way? Or do they come back?
Yes. Okay. So I've had a lot of people in my lab coming, especially from China and Japan. Yes, I'm totally aware in their career that they need to have this big nature stamp and cell, and we are working. We've published in those, and about 80% of my lab members have jobs back home. So I'm aware of that. I would hope that also the countries start to change a bit their mindset, the understanding that impact factor means nothing, that the big journals are not warrant of quality in any ways, and that you can't evaluate a career counting beans on number of first offer papers. That does not work. We have enough experience as recruiters to see that when you recruit people and you really interview a person using a chalk talk, grilling the person for 1.5 hours with no slides. We've seen people with CVs like that failing in the most terrible manner.
I'll extend this a little bit. For instance, when we are screening applications for positions, there is a screening position where the publication count is used.
Yes.
And then they don't even get to the stage that you're talking about in the interview. So I've asked my students to put up their content as bioarchive, but my students are going out to Europe and the US for a postdoc, whereas yours are coming back. So I just want to know whether there's a difference over there. Simply the bioarchive, the idea of putting up your data before it is published, and is that good enough?
Yeah, but that's good enough. That's the essence of science, is that the data is available. That's why bioarchive was made. And bioRxiv actually counts as published currently. So you do not, I mean, I've been part of EMBO LTF. You can have people who have 1 or 2 first offer in bioRxiv. That's fine. So there's a need from the agencies to move away from assessing the CV based on publication. We can't go on with this. That's unfair. That's unfair, and that represents basically mostly the past and the environment of a person, but not the person. And I know it's a big deal, and I know it's complicated when you have to deal with large numbers. I know, it's not simple.
Right. We have a networking mix, so this conversation can go on for another couple of hours. All right, thank you, Frederic.
Thank
we'll
you.
Stop. Few things. Thank you. A few wrap-up announcements for day 1. First things first, this might be the only time in the conference that you are encouraged to take out your phones. Please take out your phones, scan the QR code and give us feedback for day 1. We take this very seriously. We would like to iterate that process, the programs at EM. And so please quickly do this. Thank you, and you're filling that out. You got the link, right, through the QR code, so then you can take your time to